everyone, and welcome to today's webinar titled Optimizing Clone Selection in Cell Line Development. This webinar is part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Shelley Mulock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to sartorius.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Daryl Cole, PhD, who is an application scientist at Sartorius. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Dr. Cole, you may now begin your presentation.
Hello and welcome to this webinar entitled Optimizing Clone Selection in Cell Line Development. My name is Daryl Cole and I'm an application scientist at the Bioanalytics department in Royston, UK. So let's begin with the agenda for today. So I will start with an overview of cell line development, the actual workflow involved, and then I'll move on to a bit of information about the iQue advanced flow cytometer, which you can see on the right-hand side of the slide there. Then I'll go into a little bit of detail about a couple of the kits you can get for the iQue, starting with the cell count and viability kit, and looking at a couple of case studies for each of these, so beginning with monitoring cell culture using the cell count and viability kit, then move on to the fixable viability dyes and look at characterization of a range of intracellular markers in adherent and non-adherent cell lines, and then an extrapolation of that with a combination of both intracellular and surface marker staining in adherent and non-adherent cell lines, and finish with a conclusion and a summary of everything that we've learned today. So let's begin with an overview of cell line development, looking at the workflows involved. So if we look at cell line development workflow, we can see that there are 5 key stages of cell line development workflows. What we have is beginning with the gene cloning and initial clone selection, and then moving on from there, we go to the clone selection and confirmatory analytics step. Moving on from there, we begin with the cultivation and media optimization, so optimizing the culture conditions for your particular clones of interest. Then we look at evaluation and characterization, ensuring that whatever product your clones are producing is the correct one that you want for your studies. And then finally, we will bank those cells so that they can be used further downstream or even as a product that can be sold to customers. So what are some of the challenges involved with cell line development? So the biologics commercialization is a very lengthy process, so cell line development can take up to 12 to 18 months. So that is obviously a problem and can be a bit of a challenge. There's also a very real need to reduce cost, resources, and the time to market, as explained. So maximizing your product yield and process robustness. Then we also have data-driven decisions, so we need to be able to enable informed choices and reduce risk for later stages in commercialization processes. So some of the bottlenecks in cell line development are screening these clones in large numbers to identify your best performers, preparing and analyzing the samples, culturing clones in multi-well plates, flasks and bioreactors, and then finally all of that data you've got to process and interpret in some fashion. So that's another bottleneck. So how do we select the best producing cell lines based on comprehensive, robust and relevant data? Well, this is where the iQue advanced flow cytometry platform comes in. So this system can find the best clones, screen critical quality attributes, optimize culture conditions and characterize lead clones. So how do I select the best performing clones? That is the initial challenge here. Well, with the iQue, we can fully evaluate the ability of lead candidates, cell health and growth data. So let's take a look at the iQue advanced flow cytometer in a bit more detail. So on the left-hand side of the slide, you can see the iQue3 advanced flow cytometer. So this is what the system actually looks like. This is how it would look in your lab sitting on one of your benches. So I'll go through the capabilities and advantages of this system now, starting with one of the headline features, which is the speed of the system. It provides the fastest plate processing on the market, which enables shorter assay development, fast analysis and rapid user throughput. Additionally, the system also allows for no-wash assays, further increasing the speed of analysis. So partly the reason for this speed is the unique air gap technology that the iQue3 utilizes. So this allows you to read multiple wells within the same plate without having to do wash steps in between. The system uses an air gap between each of the samples that it takes up, and so you can run very large numbers of samples in very short periods of time. So as an example, the 96 well plate can be run in around 10 minutes, and a 384 well plate can be run from anywhere from 20 to 30 minutes. So it's a lot faster than the majority of other systems out there in the market. The system also provides rich insight into your samples and gives the user per cell resolution, 3D multiplexing, phenotyping, and also functional analysis, with the added flexibility of being able to measure not only cells, but also secreted proteins and other factors within the same well. Another key advantage is the miniaturization of the system. So you can do more with less. So the unique process of the iQue system, using a revolutionary air gap sampling process, as I've described previously, translates to smaller assay volumes. So it allows the conservation of precious cells and expensive reagents, leading to the capability to run more assays, providing more biological insight. So all of these advantages will be useful without high quality user interfacing and software analysis tools, and the iQue system excels in this key area. The Foresight software is loved by users for its power and simplicity and provides one interface from experimental design all the way through to analysis. There are no optical adjustments required, and the system has automated cleaning and calibration capabilities, taking the hassle out of flow cytometry. Finally, collecting and analyzing this data would be of no use without insight and understanding. Because of the key characteristics already described, the iQue system, in combination with the Foresight software suite, allows the user to link information, run scenarios, create knowledge, and make decisions quickly and confidently, empowering our users with the tools they need to succeed. So on the right side of the slide, you can see the three pillars of the iQue advanced flow cytometry system. So the machine itself, then in the middle, the Foresight design and analysis software, and then on the far right, the kits and consumables that provide easy to use analysis reagents and tools for your experiments. If we look below, we can also see the key areas of investigation provided by our iQue kits and consumables portfolio. These range from immuno-oncology, a range of T-cell analysis kits, to antibody discovery in the form of type and titer and antibody internalization reagents, to our foundational cell functions encompassing apoptosis, cell proliferation, encoder dyes, and finally cell count and viability agents and dyes, of which I'll be talking about today. More specifically, the cell count and viability kit and the fixable viability dyes. So let's take a closer look at the cell count and viability kit. So when it comes to measuring cell count and viability, traditionally most labs have been using the low-tech method called the hemocytometer. So I'm sure plenty of you have experience using one of these devices. Essentially, it's a glass slide with markings on it so that you can place your cells of interest on there and use a microscope to then look at the cells and count those manually. There are newer alternatives, such as the SEDEX automated cell counter, which uses tubes. So you put your samples in tubes and then the system will do the counting for you. And then there's also the ViCell viability analyzer, which also uses tubes but can also use 96 well plates as well. Each of these technologies uses the trypan blue stain to measure viability in addition to the cell counts. The iQue cell count and viability kit uses non-toxic dyes, unlike trypan blue, to analyze cell viability. So that is an additional toxic chemical that you can remove from your lab if you use the iQue cell count and viability kit. So let's have a look at the cell count and viability kit in a little bit more detail and look at the advantages that it provides the user. So the advantages here are that it's got an increased accuracy and reproducibility. It allows for high throughput analysis, so you can use 96 well plates and 384 well plates. You can save precious samples because the iQue only needs around 5 microliters of sample in order to analyze your samples. And you can therefore maximize productivity using much less sample and using the high throughput analysis where you can run multiple samples in a very short space of time. So let's take a more detailed look at the cell line development and the cell count and viability kit. So here we have the iQue3, and if we look at the steps in cell line development, we've got vector cloning and transfection, which leads to mini pool generation and confluence measurement. And then if we look at pool evaluation, single cell cloning, and clone evaluation, and these three sections utilize the iQue3 in order to analyze your cells using the cell count and viability kit. So it's critical to have a reliable solution during cell line development because the decisions that you make at pool and clone selection impact the downstream product. So if you use the iQue in combination with the cell count and viability kit, this allows calculation of specific productivity of each clone during this process. So if we use the iQue during CLD, we can vastly increase clone analysis speed, where the measurement of 96 samples can be performed in less than 20 minutes. So if we take a look at the cell count and viability workflow, so the cell count and viability kit facilitates fast and robust cell health and count analysis. And so if we just have a little look at this workflow, you can give you an idea of the kinds of hands-on time you'll be looking at when you're working with this kit in the lab. So what we have here at the start, we've got your cells, and then you have to dilute the viability dye to a working stock. So that's usually 1 in 250 in media. And then what you would do is combine your cells and the working stock of viability dye at 1:1, so 10 microliters and 10 microliters in V-bottom assay plates and incubate for 10 to 30 minutes. So what that would look like inside the well, would be your cells here. So you'd have your cells with intact membranes that would be your live cells, and then you'd have some cells with permeabilized membranes. And so the live cells would remain unstained because the membrane integrity dye could not enter into the cells, whereas the dead cells would have the dye enter through their membrane and become stained. And so you'd be able to discern those using the iQue. So if we move to the next step, step 2, we then add 10 microliters of the counting beads per well, give the plate a quick spin to ensure the volume is at the bottom of the plate. And then what we would see inside the well would be the cells again, but then you'd have your beads in there as well. And the iQue would use these beads to work out the cell density. And that's how you get your count. You got your counting beads there. And then finally, we would then analyze on the iQue platform and acquire our data with less than 20 minutes for a 96 well plate, a full 96 well plate, and then less than 40 minutes for a full 384 well plate. So if we take a look at the gating strategy, which we use with the iQue Foresight software, you can see that it's quite simplified analysis because every kit that you purchase comes with the gating strategy and everything else already laid out for you. So the provided template comes with preset gating, acquisition settings, and the analysis. And that will be imported into the iQue Foresight software. So when you want to quantitate your cell count and viability, a lot of the foundational work has already been done for you. So as you can see here on the left-hand side, we've got all events here. And what you can see is we can quite clearly separate out the counting beads and actually our cell populations as well. And then if we look at all of our cells, on the right-hand side, we can separate out our live cells from the dead based on the viability dye staining. So the data that we are able to produce with this kit is reproducible and accurate, and this counts across a wide range as well. So we've compared here the vi-cell viability counts with our cell count and viability for percentage viability using a mix of growing and heat-killed cells tested at various ratios. And what we can see here is that there's quite a linear correlation between the two methodologies. And again, if we look at the cell density calculations here with UrCats at a density comparison of 8 times 10 to the 4 all the way up to 2 times 10 to the 7 cells per mil, we can see again it's a very linear correlation, suggesting that the cell count and viability kit is accurate across a wide range. So another thing that we can look at with the iQue Foresight is the heat maps and also some profile maps that we can use to look at intuitive, actionable data. So on the left-hand side here, we have the cell density and viability of all of our cells in the wells of a 96-well plate. So these are CHO-S cells that were plated in a 96-well plate for count and viability analysis during a limited or limiting dilution cloning experiment. So these plate maps were rendered in iQue Foresight, and they show, as I've said, the calculated cell density and the viability of each of the cells. And you can see that we have different colors for the different levels of viability and cell densities as well. What's interesting is the profile map that we can produce in Foresight, which allows you to threshold for specific characteristics of your cell population. So on the right-hand side here, we've got a key set of criteria where we have the cell density locked at 0.3 times 10 to the 6 per mil, so 300,000 cells per mil, and then we are moving the viability range on the right-hand side. So as you can see, as we reduce or increase the viability, we increase the viability threshold, we see a dropping off of the number of wells. So we can quite easily, through this kind of analysis, identify those wells that have the criteria that we want, so that cell density and then a higher viability as well. So it's a very useful piece of analysis within the software. So let's take a look at the fixable viability dyes now. So these analyze cell health, allowing downstream characterization, but in this case, it's those characterizations that require fixing, such as intracellular staining. So they work in a similar way to the cell membrane integrity dye. However, these are still viable after fixing. So this allows for high throughput analysis in 96 and 384 well plates. It's fast. You can stain your cells in 30 minutes without any washing. And you can, again, use very low sample volumes, as little as 10 microliters per sample required. So this reduces attrition for downstream applications. So the case study for this particular kit, as I mentioned in the agenda, is to characterize intracellular markers. So we've used the iQue platform to make intracellular staining fast, simple, and robust. We've got a flexible assay, so we have 3 color options that provide flexibility to use a variety of conjugated antibodies in combination with fixable viability dyes. So we have a V blue, a B green, and an R red. So that gives you a lot of options to use multiple different types of fluorophores when you're looking at performing intracellular staining. So let's have a look at the workflow here for this particular kit. So if you were looking at just staining your cells for viability, you could seed your cells in a 96-well plate, stain the cells with the fixable viability dye, and incubate at room temperature in the dark for 30 minutes, and then analyze on the iQue. So if you just wanted to look at basic cellular viability, you could perform that particular workflow. So this gives you a lot of good options for analyzing viability when cells are fixed or for other assays such as intracellular staining. So if we then wanted to look at intracellular staining in addition to using the fixable viability dyes, we could then change the workflow slightly and add a few more steps. So we do the same initial work with seeding our cells and then staining with the fixable viability dye, but then we would wash the plate and then fix our cells for 20 minutes at room temperature in the dark, wash the plate again 2 times, and then we're able to stain for intracellular markers and incubate at room temperature in the dark for 30 minutes. Then we just have to do one final wash, and then we're free to analyze on the iQue. So let's take a look at the fixable viability dyes and how they enable reliable live-dead analysis in fixed samples. So this particular example is in non-adherent cells. So these cells were seeded at 2 × 10⁴ cells per well in a 96 well plate, and then they were stained with the V-Blue fixable viability dye. So there was live dead cells that were gated in the iQue Foresight software, as you can see in a standard dot plot on the left-hand side here. So you can see that we've got 88% live cells here on the left-hand side of the population dot plot, and then on the right-hand side we've got about 12% of dead cells. In the middle we have a histogram where we've actually color-coded the live and dead cells using population gating within the Foresight software, so it can give you more of a clear distinction between those two populations. So what we have here is the live cells in yellow on the left-hand side with no viability dye staining, and then over on the right, in black, we have the dead cells that have been stained with the viability dye, which means that they have a permeable membrane, so that dye can then enter into those cells. And then on the right hand side, we have essentially the same plot as we do on the left, but we've color coded as we have done with the histograms. So again, it gives you some flexibility in terms of how you want to present your data as well. But basically what this data clearly shows is that we can quite clearly distinguish between our live and dead populations using these fixable viability dyes. So if we have another look at this particular kit, but this time using adherent cells, we can show that we can also perform the same kinds of experiments in adherent cells. So what we did here again is seeded the cells at 2 × 10⁴ per well in 96 well plates and stained with the R-red fixable viability dye this time. And then what we also did is looked at using a high percentage of live cells and then a high percentage of dead cells. So at the top, we've got a high percentage of live, and then at the bottom, we've got a high percentage of dead. And then we gated in much the same way as the previous slide with the non-adherent cell lines. So we have our live cells gated here on the left-hand side of the population dot plot, and then the dead on the right-hand side. And then we use the color coding that we can apply in the Foresight software to then make it more clearer for presenting the data. And then at the bottom we have the majority dead experiment, where you can see that there are around 67% dead cells, and then this is obviously quite easily discerned in the histograms and the actual color-coded dot plots. So in order to further validate this assay using intracellular staining, we took a collection of 4 hematopoietic cell lines with 3 intracellular markers that were sourced and tested in combination with the fixable viability dyes. So this was to further investigate the utility of fixable viability dyes within a more complex assay. And so the markers that we used were CD68, MPO and TDT, and they are expressed in different types of cells of hematopoietic cell lines. And then we have the characteristics of the 4 lines that we actually chose. So each of these markers should be differentially expressed in each one of these particular cell lines. So if we take a closer look at each of these individual markers that were described in the previous slide, you can see here that what we were able to do is look at individual plots for each of these markers and then separate those out into positive and negative populations within each dot plot. And using the color coding, we were able to highlight the positives for each of the markers in yellow and then the negatives for each of the markers in black, just for ease of readability. And the good thing about the iQue is that it's very flexible. It has a very large number of channels, so we're able to multiplex each of these markers within the same well, in addition to the fixable viability dyes. So we're able to use different fluorophores for each of the antibodies we're using to detect these markers, and we're able to multiplex all of those within the same well. So if we take a look at the marker expression for each of these particular cell lines, we can quite clearly see there's some differences here. So using the fixable viability dye to look at live cells, so all of these expression profiles for each of these cell lines is in the live cells only. So these have already been gated for live cells, and then we've looked at individual marker expression. So using this adapted assay system, we can characterize each of these particular cell lines for their specific marker expression. So if we look at THP1s, we can see that they're highly expressing in CD68 and TDT, but they have no expression of MPO. It's the same with the Urcats in the case of the TDT. Their CD68 is there, but it's quite low. But there again, there is no MPO expression. Raji cells have a similar profile to Urcats, but they have even lower CD68. And then if we look at the HL60s, they're the only one of these cell lines that expresses MPO, but they also express TDT at a high level. But then the CD68 is kind of at a midpoint between what we see in THP1s and Urcats. So these cell lines were stained with 3 antibodies in addition to the viability dye, as I explained previously, all within the same well of a 96 well plate. So if we take a look at the fixable viability dyes and intracellular staining, again, we can also look at this in adherent cell lines. So for this particular case study, we used induced pluripotent stem cells. So I'll go give a brief overview of what iPSCs are. So stem cells are unique within the body. They have an ability to differentiate into multiple lineages depending on their type. They're a very important research and clinical tool, and because of their ability to differentiate into multiple different lineages, they can be used to develop key models of normal and diseased tissues. So if you're able to get a hold of a diseased iPSC cell line, you can then create disease models from that by differentiating those stem cells into whichever particular tissue type you're interested in looking at. So iPSCs themselves, they are actually stem cells that have been derived from normal tissue by being induced through expression of transcription factors. So in 2006, Shinya Yamanaka discovered this, and he used OCT4, SOX2, KLF, and c-MYC. And these were the transcription factors that were forcibly activated in these primary fibroblast cells in order to reprogram them into pluripotent stem cells. The sendovirus is the most established method that's used, but there are also episomal methods, lentiviral and retroviral, and also transfection can also be used. So what this creates is the epigenetically activated cell population, which can then be pushed further down towards an iPSC, and then the iPSCs can be differentiated into all manner of different cell lineages. So using the fixable viability dyes, intracellular staining, and iPSCs, as mentioned before, the iPSCs are useful in both research and clinical settings as described, and because the creation of iPSCs is inefficient, expensive, and time-consuming, fixable viability dyes combined with intracellular marker staining provide a robust and high throughput method for characterizing iPSCs during CLD processes. So you could look at phenotypic markers for pluripotency as an example, and further validation by combining the above assay with surface marker staining would also elucidate more insight into the cells being developed. So here we can see we can look at intracellular staining as described previously with the non-adherent cell lines, but then we can also look at surface marker staining as well. So the iQue provides facility for simple, fast and robust characterization of iPSCs or any other cell or cell line that you want to look at. So what did we actually do when we performed this case study? Well, we looked at 2 intracellular markers. So there's OCT3/4 and SOX2. These are both markers of pluripotency. So these will tell you whether or not you have iPSCs. And then we also looked at 3 surface markers. SSEA1, which is a normal cell marker, so it's a marker for non-pluripotent cells. And then we have SSEA4 and 12160, which are another 2 pluripotency markers. And we then use the fixable viability dye R-red as our live dead stain. We used a THP1 cell as our normal cell control and looked at expression of iPSC associated intracellular markers SOX2 and OCT3/4 in iPSCs. And we found that we only saw these intracellular markers in our iPSCs. there was no expression in the THP1. So if we look on the right-hand side here, these histograms that we have, we've got the THP1s in yellow and the iPSCs in black. And the top one is showing you the expression of SOX2, and the bottom one is OCT3/4. So what we can see here is in black, we have a clear shift towards the right from the THP1s for both of these markers, which is indicative of the iPSCs expressing these pluripotency markers, whereas the THP1s are further to the left, so do not express those particular markers of interest. On the right-hand side, we have the dot plots associated with these this histogram data, and you can see here that again it's the same result. We have this nice shift over to the right for our iPSCs, and these histograms and dot plots were gated from live cells in the iQue Foresight software. So if we then look at the surface marker staining, again, we've got the THP1s here as the normal cell control in yellow. And what we can see in the first histogram and dot plot is that the THP1s have high levels of the SSEA1, which is the marker for non-pluripotency, whereas the iPSCs do not express. If we move across to the two pluripotency markers, we can see that the THP1s are very low. So indicative of not expressing that particular marker, and then the iPSCs in black are shifted way across to the right, which means that they are expressing that particular marker of pluripotency. Here is another way of presenting that data. So what we have on the left-hand side is a heat map showing you the THP1s and the iPSCs, and then all 5 of the different markers: SSEA1, SSEA4, Trim60, SOX2, and OCT3/4. SSEA1 again is the normal marker for non-pluripotent cells, so we can see a high expression in THP1s, but then all of the pluripotency markers are not expressed at all in the THP1s. And then it's an inverse expression for the iPSCs, where we see no expression of SSEA1, but then high expression of all of the pluripotency markers. Moving on to the right-hand side, this is the same data just shown in a graph form, where we can pick out the surface markers and the intracellular markers. And again, we have the THP1s in yellow, so we can see a high expression of SSEA1, the non-pluripotency marker, and then no expression of the pluripotency markers in THP1s. And again, it's the inverse with the iPSCs. There's no expression of SSEA1, the iPSCs are in black, but then they are highly expressing pluripotency markers SSEA4, Trim60, SOX2, and OCT3/4, and they all correlate with the surface and intracellular markers. So to conclude, the iQue advanced flow cytometer can be used to monitor cell count, viability, and characterize intracellular phenotype during biological workflows, such as cell line development, as we've talked about. These techniques can be adapted to provide solutions for viability, intracellular and surface marker staining simultaneously in a single well via multiplexing. The integrated iQue Foresight software makes analysis of complex data fast and seamless, which dramatically reduces your time to results. So if you require any further information, you can go to any of the following web pages here. So we have some sections on cell count and viability, the fixable viability dyes, and also a technical note on intracellular staining to give you an idea of a bit more detail of how we performed the intracellular staining that we just showed on the webinar in those case studies. If you have any questions, please save them for the Q&A session, which will be just after this webinar. Thanks for your time, and I look forward to answering your questions in the Q&A session after.
Thank you so much, Dr. Cole, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located on the far left of your screen. So let's get started. Okay, so it looks like our first question is, can the cell count and viability kit be used with adherent cell lines?
So we do not provide a protocol or assay specifically for adherent cell lines, but as long as the cells have been detached prior to the staining protocol, then it would be possible to use adherent cell lines.
Okay, wonderful. It looks like our next question is it possible for the user to adjust the gating strategy found in the template for this kit? For example, if the kit cell lines used did not fit the template gates.
Yeah, the gates are completely adjustable by the user. So their placement is really a guide when you install the template onto your system. And so they often won't match up perfectly for the specific cell lines you're using. So you're able to adjust them for specific cell lines.
Oh, great. Okay, excellent. The next question is, do the fixable viability dyes show decreased signal after fixation compared to non-fixed dyes?
The fixable viability dyes have been tested quite extensively by us. So what we found is that there's no significant difference between the dye's signal before and after fixation.
Okay, good to know. Wonderful. Let's see. Our next question is, could fixable viability dyes be used in co-culture experiments with multiple cell lines per well, both adherent and not adherent?
Yeah, that would certainly be possible by making use of the iQue cell encoding dyes. So we have a range of colors for those. So you can stain your different lines with varying intensities of dye, and that allows you to distinguish them via fluorescent screening on the iQue system.
Okay, wonderful. And it looks like we have one more question, which is, could the kind of study you have performed in iPSCs be used to measure markers in differentiation during a directed differentiation of iPSCs to a specific tissue type, like say neuronal tissue?
So yeah, that is possible. You can build a panel that would be compatible on the iQue, then you can do this. So you could measure marker expression on the cells and inside the cells themselves, and then measure changes in expression over time in differentiating cells. We do actually have an app note on our website where we have actually done this. So that's there as a possible resource to actually look at on the Sartorius website.
Okay, wonderful. So thank you, Dr. Cole. Do you have any final comments for our audience today?
I'd just like to say thank you for having me. And as I said previously, if there's any other information that people want to access, our website has got loads of data and information on there for anything spoken about in this webinar, and a lot of other things as well related to biological assays.
Perfect. Okay. Well, thank you again, Dr. Cole, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Sartorius, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. The webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, goodbye.